Hi viewers, welcome to Isife EDUBLOG. My name is Kopi, and we'll be looking at this interesting topic, excretion and the excretory system. And then if you are new in this channel, don't forget to hit the subscription button to get our new or recent video. Okay. We're going to start with our lesson objective, what we need to know at the end of this video. At least at the end of the video, you should be able to define excretion and then know the excretory structures, the different types of excretory structures, and then structure of the kidney, units of the kidney, that is the nephron, then how urine is formed, urine formation, and then we'll look at a past question, and as if you hit on the past questions, you get the WAEC, NECO, JAB, and post UTME questions solved. Okay, we're going to start with the objective number 1. So we are going to look at the definition of excretion. What is excretion? Okay, excretion is the removal of waste product of metabolism. Okay, and then this term metabolism is the sum total of all the biochemical reaction. That's metabolism. There are two types of metabolism. Two types of metabolism. Metabolism can be classified into two types. We have catabolism and then we have anabolism. Okay. An aspect of metabolism that involves breakdown of molecules that are complex, the splitting or the breakdown of complex molecule into simpler molecule is known as catabolism. Example of catabolic process can be digestion. Digestion is a catabolic process. Also, respiration is also a catabolic process. Okay, breakdown of protein into amino acid or breakdown of starch into glucose are forms of catabolic process. Then in respiration, the oxidation of glucose to yield some byproducts like carbon dioxide, water, and energy. Energy is not a byproduct, energy is a very important precursor for which most cells use to generate some energy to cells. And then respiration or the oxidation of glucose or food substance will release products like CO2 and water. Release CO2 and water. So excretion is the removal of waste product of metabolism. So any waste product of metabolism called metabolites. These are metabolites. Metabolite is a waste product of metabolism. So the removal of waste products, any form of biochemical reaction that has happened to release such substance as waste is excretion. Or, on the other hand, we can also define excretion as the removal of nitrogenous waste. Another way to define it, can still define it as, I can also say that excretion can also be defined as the removal of nitrogenous wastes. Okay, the key thing there are just removed metabolism to be replaced by nitrogenous wastes. This is what I have now. nitrogenous waste. The removal of nitrogenous waste can also be referred to as excretion. This nitrogenous waste is basically from the digestion of protein. Let's say that protein is actually broken down in the cell. The digestion of protein will give you that the end product here is amino acid. Amino acid. Amino acid, which contains an alkyl group, an amine, a hydrogen atom, and a carboxylic group. This is amino acid. Okay, now, protein is an organic compound, just like carbohydrates is an organic compound. All this is under digestion. Digestion, the two aspects of food that's via digestion, they are broken down is protein, and then carbohydrates. Yes, carbohydrates. It's converted to glucose, and this is converted to amino acid. But the body never stores amino acid, but the body can store glucose as glycogen in animal. So this is the storage form of glucose. But excess amino acid after digestion is never stored in any form. That is amino acid. The reason why excess amino acid is never stored in the body is because of this group. This just particularly this, and this is called in organic chemistry. This is called amine. Amine group in organic chemistry. But here we just refer to it as amine. That is the nitrogenous group, because it's a nitrogen group. And it's very toxic. That is the reason why amino acid, which is the end product of protein digestion, is never stored in the body because of this amine. Okay, because of this amine, it is not stored. So, the liver has a very vital role to play in the process of deamination. Deamination. Yes, deamination, just like this is amine. And then if I have it as D, amine, this is amine, or I can have it as deamination. Okay, the prefix D here means removal or breakdown. Okay, just like amination, opposite of deamination is amination. So amination is the removal. Okay, the remove is the opposite of amination. Deamination is opposite of amination. So deamination is the aspect that involves the removal, just the removal of this amine, since it is toxic. One important feature of a excretory system is that excretion helps to get rid of those substances that can increase the level of toxicity in the body, or it can also give an impression by neutralizing some of these substances that are harmful, by rendering them unharmful in the body. And one of them is amine. So the liver, not in the aspect of detoxification, but deamination, by removing this very toxic part of amino acid. That is the reason why amino acid is not stored, is because of the amine. So removing this amine, a process called deamination. That's why we refer to the liver as an organ of deamination. Most examiners, most exam bodies do ask this kind of question: where does deamination take place, or where does deamination occur? Deamination takes place in the liver. What is deamination? Deamination simply means the removal of amine, or the removal of nitrogenous group, because this is still nitrogenous group. But in this aspect of excretion, it becomes a waste. I'm going to explain the series of reactions that makes it a waste product in the aspect of excretion. Okay, but in this aspect, deamination is the removal or breakdown of the amine group or nitrogenous group of amino acid. That is deamination. And the deaminating organ, the organ responsible for this deaminating process, or deamination process, is the liver, not actually the kidney that remove this amine. The role of the kidney is to excrete, not to remove. So the aspect of removal of this amine from this amino group is called deamination. So this is what we refer to as the amine. But in certain kind of organism, once this happens, once this deamination has happened, and then this is the nitrogen group here, NH2, the cut out from the amino group, the NH2, which I can give it two names. I can either call it nitrogenous, nitrogen group, or I can call it amine. So the removal of this is deamination. So we have just been able to remove it from here. And having removed it from here, that means that the other fragment here, remember that this R here can be CH3, still a hydrocarbon. This R means hydrocarbon. That's that the remaining carbon fragments, some fragments remaining here is hydrogen, is carbon, and then is oxygen. So the remaining fragment here in amino acid, when this is deaminated or removed from amino acid, so the remaining fragment is now carbon, hydrogen, and the oxygen. These are basically converted. This fragment here remaining after this removal, the remaining carbon atom fragment, hydrogen atom fragment, and oxygen atom fragment, these three elements are converted to glycogen or carbohydrates. They are both converted to carbohydrates. So sometimes in this video I'm going to tell you why sometimes protein can also give energy. You know that most times we always give that function of carbohydrate. That carbohydrate is the energy giving food. One of the functions of carbohydrates is energy giving food. But without knowing, in this video I'm going to tell you that protein also is energy giving food. But in this process, protein can give energy in the absence of carbohydrate. If the body does not have enough protein, it does not have enough carbohydrate, but there is enough protein. Now, when this deamination process happens, now the liver converts this carbon fragment. When I say carbon fragment, it's not just carbon, but another element there apart from carbon, like oxygen and hydrogen. This fragment, the same liver convert these fragments of amino acid to carbohydrate. The process of conversion of this carbon fragments to carbohydrate is called, this is a pathway of reaction. So the process of converting a non-sugar to sugar, protein is a non-sugar. Now there has been a conversion of a protein, which is a non-sugar, to now a sugar, a carbohydrate. It's called gluconeogenesis. It will be gluconeogenesis. Yes. What is gluconeogenesis? Gluconeogenesis is the synthesis of carbohydrates. Okay. I'll just write this somewhere. Gluconeogenesis. Genesis. The synthesis of carbohydrates from non-carbohydrates. Okay, the synthesis of carbohydrates. Carbohydrates is actually synthesized, but not from the main carbohydrates, but from protein. Okay, that is gluconeogenesis. So it happens after deamination. Now, now these carbohydrates, later it is stored. These carbohydrates formed after this process of gluconeogenesis is converted and stored as glycogen. Okay, it's stored as glycogen. So that at a point where there is a depletion, if there is low level of sugar, glucose in the bloodstream, okay, this storage carbohydrate called glycogen is converted, okay, is converted back to glucose. glucose. Okay, this particular step happens if glucose level in the bloodstreams are very low or depleted. So at that point, this glycogen store is converted to glucose. And then this glucose is now later oxidized to release what? Energy during respiration. That is why when you are asked to define respiration, what is respiration? We now start to say that respiration is oxidation of food substance. It's not necessarily oxidation of glucose. And even though glucose can be oxidized, other food substances like fat and oil can also be oxidized. So it is oxidation of food substance. And this particular reaction occurs in the mitochondrion of a cell prior to release of energy. And this energy are expended or cells spend this energy in the form of ATP. The ATP means adenosine triphosphate. But we're going to discuss that in another video, a short video that we have been releasing very soon on respiratory system. But aside from that, what happened to this glucose? Glucose will undergo oxidation, either it's anaerobic or aerobic. But the fact remains that this glucose is unstable, so it should be used to supply enough energy to tissues or cell. So in this case, this is what we started from here, proteins. From proteins to amino acid, from protein to amino acid, what happened from here to here is what we call digestion. So after digestion, this digestion is also a catabolic process. After digestion, then the protein here is converted to amino acid. Amino acid is the end product of protein digestion. So this protein, just like carbohydrates, after digestion, carbohydrate, which is starch, is converted to glucose. Then excess glucose is stored as glycogen. But excess amino acid is not stored in any form because of this amine that is attached to this group. So the liver being an organ that detoxify or deaminate, tend to remove this particular group that can constitute toxic to the cell. This particular group are removed. Which group are removed there? The amine. Now, the process of removal of this amine is what we refer to as deamination. So once these amines are removed, I'm just keeping it as check here. Then what happens to the remaining? Because I told you that protein is an organic compound that contains 4 elements, which are hydrogen, oxygen, carbon, and then nitrogen. These are the main elements contained in protein. Out of these 4 elements, nitrogen is among them very toxic. So the body cannot store this group because of nitrogen. So every effort is made by the cells of the liver to get rid of this nitrogen in the process called deamination. So after the deamination, the remaining carbon fragment, which is carbon, hydrogen, and oxygen, are then converted to carbohydrates. The process of this conversion of this protein to carbohydrates, process of converting or synthesizing, making, this word synthesis means making or manufacturing carbohydrate from something that is not carbohydrate, is called gluconeogenesis. After the glucogenesis, the carbohydrate so formed is converted to what? Storage form of what? Carbohydrate, which is called what? Glycogen. And at a point happen when this glycogen is converted, glycogen will remain here in storage form in the liver until when the level of glucose in the bloodstream is very low or depleted. At that point, this cell of the liver, with the help of hormonal regulation, that is a hormone that will trigger the conversion of glycogen to glucose, is called glucagon. Okay, glucagon is a hormone that is made in the alpha cell of the pancreas. Its role is being stimulated each time the blood sugar is low. The level of sugar, when the level of sugar is low in the bloodstream, glucagon is triggered to convert storage glycogen to glucose. Now, because glycogen is a complex carbohydrate, glucose is a simple carbohydrate, a simple sugar. So now, a process of this breakdown of glycogen to glucose, and this particular reaction takes place whenever glucose level in the bloodstream is too low. So the conversion of glycogen to glucose, when this happened, is called glucogenolysis. Yes, the conversion of glycogen now, just from here to here, okay, yeah, but from here to here is gluconeogenesis, but conversion of glycogen to glucose is called glucogenolysis. 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 Okay, the word lysis is here that is ending the genolysis here simply means it's a breakdown. This process is a breakdown of glycogen to glucose. Yes, breakdown of glycogen to glucose is called glucogenolysis. Okay, this is what happened to this aspect of amino acid. And then what happened to this aspect of this amide? What let's happen to amide? Okay, what let's happen to amide will depend on different kind of organism. Okay, it depends on the organism. Yes. Okay, I'm going to write this as now. It's going to be a waste now as nitrogenous wastes. It depends on the organism. Okay, I'm going to be discussing 3 organisms here that are going to be removing this waste differently. Okay, 3 organisms are going to remove this waste differently. One of the organisms that will remove this waste differently will be mammals. Mammals remove this waste differently, and then insects or reptiles or birds will remove this waste differently. And then another class of organism that will remove this waste differently will be protozoa. Okay, with protozoa. Okay, this nitrogenous waste so formed from here. In mammals, this waste is removed as urea. In insects, this waste is removed as uric acid. That's insect, reptiles, and birds. And then in protozoa, this waste is removed as ammonia. Okay, I think in this video it's very important I explain why these organisms remove this waste in this form. These 3 wastes, nitrogen waste, are not the same because basically this urea is liquid. This uric acid is semi-solid, and this is gas. Okay, now the reason why mammals remove nitrogenous wastes as urea is because terrestrial organism, when we say terrestrial organism or land organism, we mean organisms that are basically a land animal. Terrestrial organism which does not have problem of water conservation, or terrestrial organism that water conservation is not a major problem. They don't have problem of conserving water. Whether they conserve or they don't conserve, it's not their problem. So any organism that conserving water is not a problem, and this organism happens to be a land animal, and this organism does not have problem of water conservation. Basically, the form of wastes or the form of nitrogenous waste removed by such organism basically will be urea. And this example is mammals. Mammals are organisms that they are online. They do not have problem of conserving water. So because they do not have problem of water conservation, they don't have any other, they can only remove their wastes or their nitrogenous waste as urea, which is a large liquid. Because not when they remove this liquid or this urea in excess quantity, it will affect the organism. Since they do not have problem of, they remove this large liquid waste from their cell, it will not affect the cell, or it will not create another kind of concentration to the cell, because water conservation has never been their problem. Not for this kind of organism, insects, reptiles, and birds, that basically they are all terrestrial or land animal, who also face problem of conserving water. Yes, these 2 particular class of organisms are quite different. The difference in this organism is even as much as they are land animals. But because reptiles, birds, and insects do have problem of water conservation, they do not need to remove their waste product in large quantity or in liquid quantity, because once it becomes a liquid quantity, that's what we're going to refer to as what urea. But because they do not have they have big problem of water conservation. They need to retain as much water as possible they can conserve, and because of that, what they remove is almost solid. That's why I say semi-solid is when it's what? Uric acid. So because they have problem, the reason why the excretory waste is uric acid, because this class of organism have problem of water conservation. And this is the reason why their waste product is semi-solid or uric what? Acid. And in the last class of organisms, which is protozoa, that's where we're going to have the lines of protozoa, paramecium and plasmodium, paramecium, amoeba. These are organisms that belong to this particular organism. These organisms do have small size compared to the environment. They are all aquatic. Since they are all aquatic, water conservation can never be their problem. But because of the size of the organism, they remove waste in almost minute quantity. Okay, and they remove waste in form of ammonia. Okay, organisms that belongs here are amoeba, paramecium, plasmodium, and so on and so forth. Basically, I'm going to show you something. In this case, we have in mammal. This is what happened in mammal. In mammal, why it is called urea is because after tissue respiration in mammal, carbon dioxide release alongside with water. The two are not allowed to mix. Because if the water and the CO2 mix, I think that should be resulting to a situation whereby a weak carbonic acid is formed. The CO2 basically combined with that is a mammal, combined with amide, NH2. This CO2 is from tissue respiration. Because you know where this is from, okay? It's going to give you NH2 brackets 2CO. There is a formula of urea. So urea is so formed. That is what happened in mammal. So it is only mammal that the nitrogen group combined with CO2 from tissue respiration to give you that formula. In the advanced level, this is a product formed from what we call ornithine cycle. But in this video, we're not going to be discussing ornithine cycle. That is that urea formed in mammal. In insects, it is not urea formed, but uric acid. In protozoa, it is ammonia. Okay, that is on the definitions of excretion. And now we go to the second objective of the lesson, which is excretory structures.